Hello, you are most welcome, ladies and gentlemen, to Real Impartation Moment on Tuesday Night Anatomy with Daniel Okwan. Now, today's section, we want to quickly go through the histological organization of the blood vessels that we have in the human body. Remember that cumulatively, yes, these blood vessels form part of the cardiovascular system. Remember that with the cardiovascular system, yes, the heart being the engine will be the pumping organ. Yes, we also have the blood, which is the river of life. Later section we look at the blood, but today we want to look at the channels through which this blood will have to flow through, and these channels are the blood vessels that we have. And remember, because these blood vessels are going to represent channels, they will be tubular organs, and as I indicated, tubular organs generally will have three tunics or three coats, namely, yes, from the lumen to the external surface, we are going to have what we call mucosa. Yes, we are going to have what we call muscularis, and then of course we are going to have what we call adventitia. Now, for the blood vessel specifically, we are going to give particular names for these areas. So that if you talk about the mucosa of the blood vessel, we are going to call it tunica intima, and I'll explain why we call it tunica intima. And then of course we are going to have the middle component being the tunica, the muscle layer being the tunica media, the middle component. That's the muscularis. Then of course you have what we call tunica adventitia. That's of course the connective tissue component. So without much ado, yes, let's quickly go through this blood vessel that we have. So remember that blood vessels, yes, as I indicated, will serve as conduits through which substances in the blood will flow to various parts of the body where they are needed. So essentially, they will act as delivery, systems. Okay, and then apart from that, they also help regulate, blood pressure. Of course, we know that, yes, if there is increased volume, then that is what we call hypervolemia. Then of course hypervolemia will lead to hypertension, increased blood pressure. So with regards to the regulation, vasodilation, and of course vasoconstriction of the lumina of these blood vessels, they will be able to help in regulation of the blood pressure. Yes, of course, you will get to know that capillaries, which are also part of the blood vessels, will ensure that at the level of tissue or the level of the cell level, what will happen is that there will be exchange of materials, and this exchange of materials will be essentially, yes, the blood giving the cells oxygen and of course nutrients, and then taking away, of course, the waste by way of, for instance, urea, by way of course, carbon dioxide, from the cells. And then of course, yes, they will help redistribute blood in response to, of course, changing needs of the body. Okay, so of course, as I indicated, vasoconstriction, there will be some areas, for instance, if there is parasympathetic activity, then of course, the blood vessels of for instance, the GIT will have to go through vasodilation so that more blood will get there, okay, to shunt a lot of blood to this area. But of course, in situations where there is sympathetic activity, where the muscles need a lot of blood, then of course we don't need a lot of blood in the, the organs of the GIT. We don't need digestion in that time. At that time, we need a lot of energy in these kind of muscles, so that of course you can actually escape that danger. Then they also ensure that they will help in regulation of body temperature. We know that the core body temperature is around 37 degrees Celsius. Okay, yes, we know that human beings are what we call warm-blooded animals. Therefore, the blood, that flows around throughout our body, okay, in that side our body, yes, it's actually having that temperature, it's warm. So that, yes, if indeed, yes, we need to conserve a lot of heat inside us, then there will be vasoconstriction around the vessels that are related to the skin, so that you don't lose a lot of the heat, because as more blood flows through the skin as a result of vasodilation, then what will happen is that you dissipate a lot of the heat, of course, through radiation. In the same vein, if there is too much body temperature, increased body temperature, then what happens is that there should be dilatation of these blood vessels to ensure that more blood flows under the skin, so that heat within it will be dissipated to the external environment. Okay, so these are some important functions that the blood vessels are going to do. So generally, what we are saying is that yes, the blood vessel is a tubular organ. Therefore, if I take a section, whether a longitudinal section or a transverse section, what will happen is that I will see a central cavity inside, and that central cavity is what we call the lumen of the blood vessel. Remember that the plural is lumina. Okay, so if we take the lumen of the blood vessel, then what will happen is that we are saying that the blood vessel is going to have 3 tunics or 3 coats. Okay, so from the lumen, what will happen is that we are going to have what we call tunica intima. Tunica intima. Now, the reason why we are going to call it tunica intima is that yes, it is intimately associated with the blood that will be flowing through it. So that's why we call it tunica intima. Okay, yes, you can also call it tunica interna, because that's the most internal component among these 3 tunics that we are talking about. Okay, now if we take this tunica interna, what will happen is that it's going to have 3 components. Actually, the tunica interna generally is going to have 3 components, and the innermost component is a single layer of squamous epithelial cells, and that's what we call endothelium over here. Then beneath the endothelium, we will have what we call a subendothelial layer of loose connective tissue. Now remember, there's a principle that beneath every epithelial lining there will be a connective tissue. So that yes, this epithelial lining being simple squamous endothelium, beneath it there will be a layer of connective tissue. That connective tissue is actually loose connective tissue, it's actually areola, and that's what we call the lamina propria. It's what we call the subendothelial layer. It's below the endothelium. Okay, so that of course there could be that kind of nourishment in there. Then beneath the subendothelial layer is another layer which we call the internal elastic lamina or internal elastic membrane. Okay, it's actually made of elastic tissue. Okay, that's internal elastic membrane. Now remember that I told you that generally for mucosa, and for that matter tunica intima or tunica interna, generally you're supposed to have 2 components. But I told you that for the GIT, for instance, the postpharyngeal gut, there was actually what we call, the muscularis mucosae. So the muscularis mucosae could be likened to what we call this internal elastic lamina or lamella. Okay, so that's what we are going to see. But let me tell you something about this, endothelium. This endothelium, yes, is very important because it's going to make, some secretions. Actually, yes, some secretions will include something like collagen, okay? So that, yes, within the smooth muscle over here, yes, the smooth muscle that we are going, the collagen fiber that we're going to see in the connective tissue, yes, could be contributed by what this endothelial cells. So the endothelial cells will be secreting collagen, mainly of course type 2, of course type 4, and of course type 5, as well as even laminin, so that it forms part of what we call the basal lamina. Okay, so that's what we are going to see. Now remember that between what we call the endothelium and the subendothelial layer will be a thin layer of a basal lamina. Okay, like the basement membrane. Of course, we know every epithelium will sit on a basement membrane, so that's what we are going to see. Okay, now even apart from that, the endothelium will be important in secreting a component which is very important as far as activating the clotting pathway is concerned, and that's what we call von Willebrand factor. Now this von Willebrand factor is needed for platelet aggregation, which is needed for platelet activation to of course cause cascade of the clotting pathway. Even apart from that, the endothelium will be important in secretion of what we call endothelin. Now, this endothelin is important. We are actually going to have the endothelin type 1. So this endothelin type 1 is very important as far as, it's a very potent hypertensive agent, actually. Then, importantly, it will also produce what we call nitric oxide. Now, nitric oxide is very important. It's a potent vasodilator. Okay, so that it causes vasodilatation to ensure that yes, more blood actually flows through the lumen of the blood vessel. So these are some things that we see regarding this endothelium. Now this endothelium could give rise to a fold which you find it exclusively in of course, the veins, as far as the blood vessels are concerned. But remember that we also find it in what we call lymphatic vessels. Okay, lymphatic vessels will also be equipped with these valves. These are folds which are derived from the endothelium. Okay, so that's what we are going to see. Now, the next one, so over here, what we are saying is that yes, the tunica intima is made up of course, endothelium, simple squamous epithelium, then of course subendothelial layer of loose connective tissue, and then a third component which is synonymous or analogous to what we call the muscularis mucosae of the postpharyngeal gut, being the internal elastic lamina. Now this internal elastic lamina therefore will serve as a boundary, okay, between of course the tunica media and the next tunic, which we call the tunica intima, and the next tunic being the tunica media. Now, tunica media, M for middle, so that's the middle tunic, and M for muscle. This muscle is actually smooth muscle. Smooth muscle is what we are going to actually see here. So tunica media made of smooth muscle. Okay, so that's what we are going to find here. Now, although yes, we are going to see smooth muscle component being there, but actually what we are going to see is that for some types of blood vessels, which we will see being elastic arteries, we are going to have a lot of elastic fibers. Okay, in here we are going to see a lot of elastic fibers with few smooth muscle cells. Okay, so that's what we are going to see. Even with the areas where we see smooth muscle, there will be some element of elastic fibers. Now, beyond this middle tunic being the tunica media, we are going to see there is another elastic lamina or elastic membrane being external elastic lamina. Now this external elastic lamina will therefore serve as the boundary, yes, between what we call the tunica media and of course the tunica adventitia. So that is what we are going to see regarding these blood vessels. Now remember that blood vessels mainly we are going to have the artery. So this is generally what we are seeing for artery. Then we are going to see that of the vein. Then intermediate we are going to see what we call the capillary. So we will see all these dynamics as we move on. Now for capillaries, where I'll come to, yes, it's not going to show this peculiar three-code system or three-tunic system, but what will happen is that it will be made up of only a single layer of squamous epithelial cells. So it's going to have an endothelium sitting on a basal lamina. So as I indicated, now this is just a cross section showing what we are talking about. Now you see this is what we call the tunica intima or tunica interna. Now this is the tunica media. Now in between the tunica intima and the tunica media, we are going to see this undulating oriented elastic fibers being that of the internal elastic lamina. And then of course, yes, beyond this one we are going to have a layer of loose connective tissue actually made up of that is the tunica adventitia. But in between, separating the tunica adventitia and the tunica media will be an external elastic membrane or external elastic lamina. Okay, so that is what we are going to see. Now, I told you that yes, veins will be endowed with valves. So that's what we are going to see. So if you see something in the lumen, okay, which will have that kind of squamous lining, epithelial cells, then of course it is derived from what we call the tunica intima, actually from the endothelium. Okay, and what it's going to do is that these valves, just like we saw in the case of the chambers of the heart, they will prevent the backflow of blood, or they will ensure unidirectional flow of blood. Now we get to know that blood that flows through veins actually flows through it under reduced pressure and therefore flows slowly. If that is so, then there's that likelihood for retrograde of the blood or the backflow of blood. Therefore, veins are endowed with these valves along their lengths so that we ensure, especially those ones which are going to move against gravity, they will have a lot of these valves. But I told you that veins will not be restricted to only valves, will not be restricted to only veins, but we also see some in the lymphatic vessels. Now, one thing that we will see is that we will come to the classification of all those, these blood vessels, being an artery, being veins, being capillaries. We see the various categories, and of all those ones will be based on the layers or the various tunics that we have. There will be that kind of variations that we will see regarding the various tunics, okay, of each blood vessel, and that will help us in identifying each vessel. But what we find is that yes, for these large arteries and of course large veins, because their wall tends to be a bit thicker, then what will happen is that yes, the blood that is flowing through them, okay, may not be able to supply the various tunics of what we call the blood vessel with nourishment. Okay, therefore what will happen is that just like we have the heart having the largest vasa vasorum in the human body, being of course the coronary arteries, yes, to supply the heart itself, although it is pumping blood to other body parts, itself requires that kind of nourishment that it is giving out. Likewise, as the blood vessels are allowing these nutrients or this blood to flow through, it also requires that it takes some of the nutrients. But what will happen is that because of how thick now these larger vessels are, of course these large arteries and of course large veins are, then what will happen is that areas, especially the distal parts or the external part of the tunica media, as well as the adventitia, will be devoid of that blood that is flowing through it. Of course, for the tunica intima and of course the internal half of what we call the tunica media, they will get that kind of nourishment because it's very closer to it. Okay, it's intimately associated with the blood that is flowing through over here. Therefore, the areas which needs attention regarding blood supply will be the adventitia as well as the external half of what we call the tunica media. Therefore, this area will have to be supplied by branches of the blood vessel itself or by branches of neighboring vessels. Okay, and that is what we call vasa vasorum. Vasa vasorum. So if you hear vasa vasorum, then we are talking about vessel of vessel. Vasa vasorum, the blood vessel, which is supplying the blood vessel itself, this larger blood vessel itself. So these vessels are, you can see they are very small, arterials, as well as some venules in there. Okay, so that's the vasa vasorum. Vasa vasorum. Okay, so we can see one here. Okay, there's another one, vasa vasorum, that we can see over there. Okay, so if we are having a vessel which is quite thicker, then we are going to find a lot of this vasa vasura in there. But what happens is that in histological sections, what we find is that we find more vasa vasura in large veins than in large arteries. And the proposed reason is that yes, because veins will be carrying oxygen poor blood or deoxygenated blood, yes, the nutrient requirement by these tunics will be less compared to that of the in the case of the artery. But if you think about it this way too, yes, that arteries would generally have much thicker wall compared to that of vein, then it makes sense that yes, we will also find these vasa vasura in arteries as well. But in most sections, we tend to find more in the large veins than in the large arteries, because large veins will be carrying deoxygenated blood. But in any case, we want to actually think through it, that yes, it could be that yes, arteries are having thicker walls, and therefore yes, it may not be surprising that in a particular section you may have more vasa vasura or vasa vasorum in that of a large artery compared to that of a large vein. So that kind of variation may be there, but generally what we know is that vasa vasorum we will find more in the tunica adventitia of large veins than in large arteries. Now there is another thing which we have to also look at it. Apart from vasa vasura, we also have what we call nevi vascularis. Nevi vascularis, also in the tunica adventitia. Now this nevi vascularis, yes, will ensure that it supplies the smooth muscles, okay, of what we call the blood vessel in the tunica media. So that of course can help in controlling its what we call contractile activity. Okay, so that is not only are we having vasa vasorum, but we also have the nevi vascularis, nerves in the vessel, the nerve which is going to supply the vessel. And this brings to another point, something we call vasa nervorum. Vasa nervorum. Now for vasa nervorum, they are actually blood vessels which supply nerves. Remember that these are nerves which supply blood vessels. That's why we call them nevi vascularis. But for vasa nervorum, they are actually vessels, blood vessels which supply neighboring nerves. Okay, neighboring nerves. actually peripheral nerves, okay, will be supplied by vasa nervorum. So we go through the various categories of blood vessels that we have, and we're going to start with the arteries. Now, arteries, yeah, we are going to have what we call the largest arteries that we have, being known as elastic arteries. Then we go to what we call muscular artery, that's the medium sized, okay, compared to that of elastic arteries. If we say elastic arteries are large, then the medium sized ones will be called muscular arteries. Even the muscular arteries, you can have the large muscular, yes, the medium size, even the small muscular arteries. And then of course you get to what we call the arterioles, which will end in what we call meta-arterioles. And then of course we will get to all those areas. Then we go to the capillaries, you will see that. So beginning with elastic arteries. Elastic arteries are the largest arteries that we have in the human body, and we can also call these elastic arteries, conducting arteries. And the reason why we are calling them conducting arteries is that, yes, they are going to conduct blood from the heart to what we call the medium-sized arteries, which we will see. Now, during cardiac systole, what will happen is that, yes, there is that kind of distension in what we call the blood vessels, these elastic arteries, okay, they stretch, okay, so you can see the lumen has become larger. And then of course when blood is leaving it, or during cardiac diastole, then what will happen is that there's that kind of recoiling of the blood vessels, such that the lumen becomes a bit smaller, as you can see over here. Now what will happen is that these elastic arteries, yes, mainly you are going to see in the case of the aorta. Then of course the major branches that we find in the arch of aorta, yes, of course brachiocephalic trunk, yes, talking about the common iliac arteries, of course subclavian arteries will also be part of it. Then of course even the the aorta will descend and then eventually bifurcate to give off what we call the left and right common iliacs. So the common iliacs are also there. Then of course you look at the pulmonary trunk, which is a great vessel. Okay, pulmonary trunk is also one of the elastic arteries that we are going to find over there. Okay, so these are some examples of elastic arteries. Okay, so that is what we see. Now for elastic arteries, yes, no doubt we are going to find what we call the tunica, intima, made up of the endothelium. And then of course, yes, we are going to have the subendothelial layer. Then of course we are going to have the internal elastic lamina. But because this one is an elastic artery, what will happen is that because it has a lot of fenestrated elastic fibers, okay, or elastic tissue in its tunica media, with little smooth muscles being there, then what we are saying is that, yes, it's going to have we don't really see or appreciate the internal elastic lamina, although it is there, but we don't really appreciate it so much. Okay, so we don't really appreciate it, so it's less conspicuous. Then even for the external elastic lamina, virtually we don't really see it. Okay, but you can actually demarcate where it's supposed to be. So one thing that is striking that we talk about regarding the conducting arteries or the elastic arteries is that the main feature will be the thick muscle layer, okay, the tunica media, having a lot of elastic fibers, fenestrated elastic fibers, okay, over here. Then one thing too is that, yes, because it is having several of these elastic fibers, then we don't really appreciate the internal and of course external elastic laminae. So of course it's going to have a very small tunica adventitia. Then of course because there's a very large artery, then it's going to have what you call the vessel of vessel, vasa vasorum, will also be present in the tunica adventitia. So that is what we are going to see regarding what we call conducting arteries. Now the next artery that we have will be the medium size, so to speak, and that's the muscular artery. Just like, yes, we saw in the case of the elastic artery, the muscular artery, yes, we also have what you call tunica intima. This one is actually a bit thicker than usual. Okay, so tunica intima will have an endothelium with, of course, simple squamous epithelium. Okay, then it will also have a subendothelial layer of loose connective tissue. Then of course beneath that we are going to have this undulating, yes, nature of elastic lamina being, of course, internal elastic lamina. So there is the endothelium, there is the tunica intima, and of course below that we are going to have tunica media. So tunica media can't be clearly distinguished. Now this tunica media is made of smooth muscle with few elastic fibers. But usually if you stain using the conventional H&E staining, yes, you don't tend to see these elastic fibers unless you stain with special elastin stains. And principally you can use the Ocean stain, okay, and then you can actually appreciate those ones. Now muscular arteries can therefore be called distributing arteries. And then these arteries are what you call named arteries, and usually they move with their corresponding veins. Okay, they are the named arteries. Now what happens is that usually for the largest muscular arteries, they will have about 30 to about 40 layers of these smooth muscles. Now remember that for elastic arteries, they are going to have about 40 to about 70 layers of what you call elastic fibers, laminated elastic or fenestrated elastic fibers. This one will have about 30 to about 40. Okay, so it means that considering the thickness of what we call the large artery being, of course, the elastic artery compared to this medium-sized artery being the muscular artery, yes, you can see there's a difference going to be between the thickness of the wall. So that is what we are going to see. Now one thing is that for the subendothelial layer, okay, for the distributing arteries, okay, for the subendothelial layer, what will happen is that this subendothelial layer, as we move on to, of course, the large muscular artery, to medium size, to, of course, to small, okay, they will begin to actually diminish. They diminish as you move downwards. Eventually, when we get to the level of arterial, they are no longer even there. We don't find a subendothelial layer in the arterial, because from the muscular artery, large muscular, yes, medium size, then it continues to diminish. Then by the time we reach what we call the arterial, it is no longer even present. Okay, that's what we are going to see. Again, what we see is that for this internal elastic lamina as well as the external elastic lamina, it tends to actually diminish as we move along the length as we move downwards. Okay, so that's what we'll be seeing. Right, so this is the muscular artery that we have. Now for the muscular artery, I told you that we have the large one, the medium sized one. Now usually for the medium sized one, they tend to move with their corresponding veins. Now remember that veins will have relatively larger lumina. Okay, we will see the differences pretty soon. Then of course they will have collapsed lumen, because of course they don't have a lot of these kind of smooth muscles to actually with elastic fibers to actually keep their lumen patent, unlike in the case of arteries. So you can see there's the internal elastic lamina that you can see over there, quite distinguished, and of course the external elastic lamina is also there. Okay, not as clear compared to that of internal elastic lamina. So there's the tunica media, the muscle layer, then of course there's the tunica adventitia. Now one thing that we know that can also help us distinguish between the artery and the vein will be that the vein, its tunica media and tunica adventitia tend to blend, because in the case of the tunica media, we have a lot of infiltration of what we call fibroblasts. Okay, fibroblasts, which we expect to be in the case, collagen fibers, which we expect to be in the case, because fibroblasts are going to secrete what we call collagen fibers, which we expect to be in the case of the tunica adventitia. We see a lot in the muscle layer, therefore it tends to blend. Okay, so that we don't see a clear-cut partition between, of course, the tunica media and tunica adventitia. Moreover, we don't even see or appreciate internal elastic lamina as well as, of course, external elastic lamina. So these are some identifying features which can actually help us distinguish between these ones. So as you can see, yes, compared to elastic arteries, what we are saying is that these muscular arteries will have decreased elastic fibers. As we said about these ones, they will have a lot of the smooth muscles. Then of course there will be thinner tunica intima. Then of course thicker relatively thicker tunica adventitia compared to that of elastic arteries. So So there we are. This, as we move progressively downwards, I told you that yes, we are going to have that of the arterial. The arterial being the smallest, artery type that we have in a human body. Okay, so the arterial, yes, we also have an endothelium of smooth of what we call simple squamous epithelium. But remember, sometimes the endothelium may be simple, cuboidal. Sometimes it could even be simple columnar. In some rare cases, you may see that. But actually, strictly speaking, endothelium is supposed to be simple squamous epithelial tissue. Then, of course, yes, we are going to have the layer of sitting on a basal lamina. We don't have subendothelial layer, but we may have, about 1 to 3, layers of what we call smooth muscle in there. 1 to 3 layers of smooth muscle in there. Then its connective tissue blends with the surrounding, structures. Then, of course, this is that of the venule. We will see venule. Now, this is a venule, just like, these muscular arteries, which will go with their corresponding veins. Arterials also will be moving with their corresponding venules. Venules are also the smallest veins in the human body. Okay, so you can see that each lumen is collapsed. This one is a bit rounded, it's patent. Then, of course, you can see that its wall is a bit, less thick compared to that of, the arterial. So for veins likewise, we are going to have light-sized veins, the medium veins, and of course, we're also going to have venules, and they will have similar organization to that of the arterial system that we have. Okay, only that there will be differences, especially at the tunica media. Tunica media of veins is less thick compared to the tunica media of arteries. Again, tunica adventitia of veins will be thicker compared to that of in the case of arteries. However, in terms of the entire thickness of the wall, from tunica intima to the tunica media and of course to tunica adventitia, the entire thickness in that of arteries, it will be thicker compared to that of veins. So there's a large vein that we have. Yes, you can see that it's going to have a well-developed tunica intima. Tunica intima, we mean that yes, endothelial layer, the endothelium will be there. There will be a subendothelial layer of loose connective tissue. Then, of course, yes, there will be a thin, which I said, be reduced. So there will be smooth muscles, okay, of what we call smooth muscle, which is also endowed with a lot of connective tissue fibers. Collagen fibers will be there with few elastic fibers, okay, forming what we call this tunica media. Then we have a very thick tunica adventitia. So tunica adventitia is well developed, okay, in this case. But as I indicated, yes, both internal and external elastic laminae are essentially inconspicuous. Now, let me tell you something about this, distributing arteries. Now, I told you about distributing arteries. Distributing arteries, which we saw, yes, we are going to see mainly they are the named arteries. Talk about the renal artery. Yes, talk about, of course, the suprarenal arteries, okay? Talk about, various arteries that we have in the human body, which are actually named, like femoral artery. So all those arteries, like brachial artery, they are named arteries. The axillary arteries, those arteries which are named, yes, they are going to be the distributing arteries. They will branch further like a tree, giving off several branches, eventually to get to the venules. The arterioles. Now for the large veins, yes, its example will be the superior vena cava, as well as, of course, the inferior vena cava will be examples of large veins. Okay, so large veins, yes, we can see these are what I have for you. Now for veins, yes, we are going to see this is actually a small sized vein. Now small sized vein, yes, is also muscular. Yes, it will be running with its corresponding artery. Now you can see that the artery is a bit thick walled compared to that of the vein. At the end of the day, they are blending to share a common tunica adventitia at the end of the day. So what we are saying is that you can see that the lumen of the vein is quite, larger compared to that of the artery. Okay, that of the artery. That's what we are going to see. But in the case, what we are saying is that veins generally will have what we call valves. Okay, so this is an example of the valve that we can see over here, where it's coming out, which we said that is going to ensure unidirectional flow of blood. Now, if you look at this is a medium-sized vein. Now, this medium-sized vein is quite thicker, okay, as we said. And we said a medium-sized vein, yes, will be moving along with corresponding, sorry, this is a medium-sized artery having a patent, okay, lumen. This is a medium-sized vein having a collapsed, lumen. It will be moving alongside with its corresponding, artery, okay. So that's what we are going to see. But what we see is that this one is a bit thick walled, okay, compared to that of the small size that we saw, okay. This is a bit thick walled. So if you are seeing that it is a bit thick walled, okay, then it tells you that it's going to be a medium sized, vein. So that is what we see. Then we are also going to see some valves, and then we said that these valves, as the veins enlarge, okay, so small size vein may not have enough valves, but medium size and of course large veins are going to be endowed with a lot of these valves. Yes, again, we want to give further description to this kind of medium sized vein. This medium-sized vein is a bit less thick walled compared to what we've seen already. Now, medium-sized veins, examples, femoral vein, yes, pyramidal centric vein. Okay, these are all medium-sized veins. Okay, and then of course, there are coats. We are going to have the tunica intima. Tunica intima being a bit thin. Okay, made up of course what we know endothelium and the subendothelial layer of loose connective tissue. Yes, of course, the tunica media being less muscular, as we said, with a lot of connective tissue fibers in there, smooth muscles, as well as collagen fibers being there. Okay, and that is what we call the tunica media. Then of course tunica adventitia. But we said that there's not going to be a clear distinction between of course the tunica adventitia and tunica media. So just to enlarge that one for you again, this is a medium-sized vein, medium-sized artery. And of course, this is a medium-sized vein. The medium-sized vein, yes, having this kind of collapsed lumen. Okay, you can see that there's blood in there. Then of course, similarly, yes, endothelium will be there. Okay, forming part of the tunica intima. Now, of course, there will be a subendothelial layer, although not clearly shown here. Then of course, we know that this will be the muscle, tunic. Okay, the muscle tunic, which blends with the connective tissue. Therefore, you can't see the clear-cut distinction. But essentially, what you have to know is that the tunica adventitia will be larger compared to that of the tunica media. Then, of course, this is the medium-sized artery because it moves with its corresponding vein. This one is going to have a thick, relatively thick, tunica adventitia. You can actually appreciate what we call the internal elastic lamina. Okay, over there, which has actually overshadowed what we call the tunica intima. The endothelium actually. So there's a muscle layer, tunica of, there's the tunica media, there's the tunica adventitia, which blends with the connective tissue, okay, of the surrounding connective tissue actually. So now let's look at the distinction between veins and arteries. Okay, so we know that if we're away, so we know that arteries will carry blood away from the heart, and then veins will do otherwise, will carry blood towards the heart. Then, if that's the case, yes, it means that arteries will distribute blood from the heart to other body parts. Then veins will carry that deoxygenated blood from the body part towards the heart. Okay, then we say that yes, arteries, the blood that will be flowing through it will flow with high pressure, or will flow through under high pressure. Veins under low pressure. Because veins are flowing and the blood is flowing through veins at low pressure, veins will be equipped with valves to ensure that we prevent the backflow of blood. Then valves will be absent in the case of artery because we don't need it. Then of course veins, except that of pulmonary veins, and then of course that's in post-fetal life. But in fetal life, as well as of course the umbilical vein, they will carry what we call deoxygenated blood. Then for arteries, arteries will usually carry oxygenated blood, except pulmonary arteries, as well as of course, in fetal life, umbilical arteries. Arteries. Then for the veins will begin from capillaries. Then arteries will actually terminate in capillaries. Okay, so you see that. Then veins, usually because blood is flowing through under low pressure, then it can be superficial, because even if there's a cut, then you don't lose great deal of blood. So veins tends to be a bit superficially placed, but arteries are deeply placed actually, because blood will be flowing through under great pressure. Therefore, if it's superficial and there's a cut and blood is actually oozing out, then you can lose a great deal of blood and death may result. So we say that veins collapse when there is no blood. Yes, veins will collapse when there is no blood. Yes, as we've also been seeing. But arteries will have patent, lumen or lumina. Then veins can further, yes, the smallest veins in the human body are known as venous. The smallest arteries in the human body are known as arterials. Then, veins are usually flattened or collapsed. Yes, we have collapsed wall. Arteries will have relatively thick wall. Yes, because veins are having less thick wall, then it means that we have a larger lumina. But arteries, because they are having relatively thick walls, then they have what we call relatively smaller lumina. Now, in atlases, what we see is that veins are labeled with a bluish color, arteries with reddish color. Okay, yes, that's what we are going to see. Then, of course, blood will flow through veins and low pressure, so they will flow through it sluggishly. But this one, there will be spurty movement of blood, in actually arteries, and that is why we can actually feel the pulse, okay, of blood flow in arteries, other than in veins. So that is what we're going to see in venous wall. Is injured, blood comes out, collects in a pool, small area around veins. But in arteries, if there's a cut, then there will be that kind of pumping of blood. Okay, a lot of blood will be coming out. So these are some differences that we find regarding arteries and veins. Now the next blood vessel, having seen arteries and veins, are the capillaries. So capillaries actually deviate from the normal trade tunic system that we have for blood vessels. For capillaries will have a single tunic, and therefore we can call that single tunic, the tunica intima. Now, even with that, it's having just one cell thick. It's just one cell layer thick. So this is the simple squamous epithelium forming the endothelial lining, and this is the lumen, okay, sitting on a basement membrane. Basement membrane is non-cellular. So that's the basal lamina. There's no cell. So we say that capillaries are one cell thick. Now, usually we find that capillaries will have what we call some cells, okay, with some actin fibers in there, and we call those cells parasites. Now, parasites can be likened to these kind of myepithelial cells that we find surrounding, the secretory units of either serous acina or mucous acina, the cells that we find that secrete these kind of secretions for glands. Okay, so that's what we see. Now these parasites would therefore help control blood flow through the blood vessels by extending their long projection. They will have some kind of long projections around the contracting or relaxing endothelial cells. Okay, so that's what they are going to do. Now, it is believed that these parasites may also contribute to the development, remodeling, as well as permeability of the vascular system at this area of the capillaries. Okay, so as I indicated, yes, capillaries will lack tunica media, and therefore they don't have any smooth muscle. If you are lacking tunica media, then no smooth muscle. They are having only tunica intima, and even with the tunica intima, it's only a single layer of what we call endothelium sitting on a basal lamina. Okay, so that's what we are going to see. Now, with regards to these two main things that capillaries will have, having a single layer of endothelial cells as well as a basal lamina, okay, by way of some modifications that we have in these two components, then we can distinguish between three types of what we call capillaries. Three types of capillaries, and these three types of capillaries are, yes, the continuous capillary. Now the continuous capillary is non-fenestrated, such that yes, the endothelial lining is intact, okay, endothelium is intact, there are no fenestrations, as well as the basal lamina is also continuous, no fenestrations or no pores in there. Okay, so no openings in the wall, yes, having a continuous basal lamina. Okay, and then the extracellular cells, okay, continuously join with each other. Okay, so that endothelial cells, there are no space in between them. That's the endothelial cells are continuously joining each other. So this one maintains a complex intercellular junction, okay, by way of some kind of proteins. okay, by some kind of proteins. So this is what we call, continuous, or that is the non-fenestrated, capillary, non-fenestrated capillary. So just like what we've seen over here, yes, we've seen in the case of the nervous tissue, the CNS, brain, yes, the lymph nodes, yes, in the muscle, in the skin, in the lungs, of course, in the heart, we are going to see these kind of continuous non-fenestrated capillaries. They also have some kind of fenestrated capillaries, although it's continuous. So, but the nomenclature continuous is telling us that the basal lamina, okay, is non-fenestrated. The basal lamina is continuous. Okay, that gives us the nomenclature continuous. But over here, this one is fenestrated. It's having, some kind of opening, some pores in between the endothelial cells. Okay, in between the endothelial cells. So this one, yeah, there are fenestrations in the endothelial cells, and then the fenestry will be covered by a small, non-membranous permeable diaphragm. So it may have diaphragms or it may not have diaphragms. Okay, so that's what we see. And then the basal lamina is actually continuous. That's a key point that we have to know. Then there's less complex intercellular junction proteins. Yes, because in between the cells, you can have a lot of these gaps in there. Areas that we're going to see them, yes, will be endowed with the most of the endocrine glands. Yes, we're going to see some in the gastrointestinal tract, especially areas where absorption is going to take place, especially the intestines. Yes, as well as, of course, the glomerulus of the kidney. We are going to see that one too there. Then we also have what we call the sinusoidal. Now, sinusoidal, we will conveniently refer to it as discontinuous capillary, because over there, apart from having the endothelial cells having larger gaps, okay, having large fenestrations in there, it is also having a discontinuous basal lamina. You can see the basal lamina is actually discontinuous, like pieces. So we are going to see that it's going to have large lumen. Actually, yes, it's going to have large gaps in between the endothelial cells, and then the gaps have no diaphragm, unlike in the case of the fenestrated capillaries where you may have diaphragm, but these ones do not have diaphragms. Then there's discontinuous or absence of basal lamina. Actually, it's discontinuous, so that's why we call it sinusoidal. We are going to see it mainly in, of course, these lymphoid organs, including the liver, the bone marrow, the spleen. Okay, we'll have what we call discontinuous capillaries. So this, showing you, under section in a different view. Okay, so this one is what we call the continuous capillary without any fenestrations. Okay, this is the basal lamina intact. The endothelial cells having, some connections by intercellular proteins connecting them. Yes, if you see this one for the fenestrated, there will be some kind of, small spaces in between endothelial cells, but still the basal lamina is intact. Then, of course, for the sinusoidal, which is discontinuous, apart from having large endothelial cells, intercellular spaces in between the endothelial cells, it's also having what we call, discontinuous basal lamina. So you can see the basal lamina is actually a lot of these spaces. So that can actually help in something like filtration, for instance, especially in the spleen. Okay, so that actually the kapha cells can get themselves lodged somewhere. The I2 cells, for instance, can get themselves in the case of the liver. Okay, for instance, can talk about the those kind of macrophages. Okay, either in the bone marrow or in the liver or wherever they find themselves, can find places to find themselves. Again, also for filtration. So this one is actually showing, yes, a muscle tissue. So muscle tissue with a cross section, you can see these are muscle cells. Okay, and then you can actually see that you can see these blood cells moving. These are actually arteries. You can see some other blood vessels. These are actually red blood cells moving in a single file. So telling you that these blood vessels, okay, having small lumina, these are what you call the capillaries, so that it can help in exchange of materials. Okay, so one thing that you have to also know is that we have more capillaries in areas where we have a lot of metabolic activity taking place. So for instance, you have the liver, the muscle, the nervous tissue. You have a lot of blood capillaries. Okay, then you have areas with less activity, including tendons, ligaments, aponeurosis. There's less activity, and therefore, of course, the blood capillaries that will be there will be a bit reduced in quantity. But we have areas where we don't have capillaries at all. They are avascular areas, and typically we are going to see the cornea, the islands, as well as even the cartilages. Okay, they will be devoid of blood capillaries. They will be avascular. So that's one thing I have to know. Now, one thing is that, yes, let's look at this. Now we know that we have the arterial system. We are going to have elastic artery. We are going to have the various muscular arteries. Yes, the large muscular, medium size, even the small. Then eventually we get to the level of arterial. And then the arterial will give rise to what we call the meta-arterial. Now, the meta means beyond. So we go beyond the arterial, we are going to get the meta-arterial. Now remember that each meta-arterial, okay, could give rise to about 10 to about 100 capillaries, okay, forming this kind of bed that we call the capillary bed. Okay, so if there's a meta-arterial, it gives off several branches. Okay, 10 to about 100 capillaries could be derived from a single meta-arterial. Now these meta-arterials, yes, you can see that we having some kind of sphincters over here, which we call them pre-capillary sphincter. Before we get to the capillary, yes, around where we have the meta-arterioles, we are going to have these kind of pre-capillary sphincters, which are rings of smooth muscles which are going to regulate blood flow through these capillaries. Okay, by so doing, they will ensure where the blood will have to go. So I'm going to tell you what you're going to see. Now these meta-arterioles that I'm talking about can either serve as what we call thoroughfare channels. So if they are thoroughfare channels, then what will happen is that, now look at this. In situations where these sphincters close, then what will happen is that the blood will not be moving into the true capillaries. Okay, so that we ensure that, yes, there's enough time for exchange of materials at the level of the tissue. So what will happen is that this kind of blood is shunted straight from the meta-arterioles when these precapillary sphincters close. So they are shunted directly into what we call the venous system. So of course into what we call the venule, and then taken into the vein eventually. So at rest where the sphincters close, then blood is shunted, and that's what we call the thoroughfare channels. Okay, these thoroughfare channels will ensure that blood is taken all the way from, of course, the what we call the meta-arterioles to the venule. Okay, but these kind of meta-arterioles can also give off branch. I've told you that the branches could, if these sphincters actually open, okay, in an active case, then the blood will get into the true capillaries, okay, with few going through what we call the thoroughfare channel through the shunt system, eventually into what we call the venule. So that if it gets into the true capillaries, then what happens is that there will be enough time for exchange of materials at the level of the capillary bed. Okay, so that is what we have to actually know. Now, one thing that we have to know, which is a take-home message, is that yes, we are going to have the conducting vessels. Now, if you talk about conducting vessels or conducting arteries, they are the elastic arteries. The distributing vessels or the distributing arteries are the muscular arteries. The resistance vessels are the arterioles. Why? Because at the level of arterioles, yes, the lumen size is actually small, and therefore, yes, less blood goes there, so that it can give enough time, okay, supply what we call the capillaries, okay, so that the turbulence of blood flow is actually reduced, becomes smooth. Okay, so the resistance vessels, okay, is what we call the arterioles. Then the thoroughfare channels representing the shunts will be the meta-arterioles. Then exchange vessels at the level where there will be exchange of materials in between the cells and the blood will be what we call the capillaries. Then of course, for capacitance vessels, for storing, capacitance has to do with storing, will be the veins. So this is what we've been talking about. Yes, we know the heart is the engine of the cardiovascular system. It pumps it blood. Yes, for instance, it gets into the aorta, being the largest artery in the human body. That's an elastic artery. Then eventually, yes, it gets into what we call the distributing arteries. Okay, the medium-sized arteries or the muscular arteries. Yes, for instance, into the femoral artery, into the renal artery, for instance. Then, of course, eventually it will branch further, giving into what we call the arterioles. Then, of course, into meta-arterioles. then of course into what we call capillaries, where exchange will take place at the level of capillary. Then of course from there into what we call the this is actually the artery. Then of course into the muscular artery, into the arterial, into the level of the capillary. Then of course into the venule. Then of course into the medium-sized vein. Then of course into what we call the large vein. So for the large vein could be that of the inferior vena cava or superior vena cava, for instance. Then of course if it is femoral vein, for instance, yes, that's a medium-sized vein. Of course back into the heart, into what we call the superior and inferior vena cava, into the right atrium of the heart. So that is, the general organization of what we call the vascular system, okay, by way of the blood vessels which we have in the human body. I hope you find it helpful, and we will move on and we'll look at actually the blood in our later section. May God richly bless you. Have a good night.